All right. Hey, everyone. I want to go through the eras with you, and it's kind of annoying. I have to redo this again, but I'll pare it down for you. The history of biotechnology is not in our textbook, which is kind of annoying. But there are three major eras that are associated to biotech. There's ancient and classical, and we are currently in the modern era for biotechnology. So there's lots of dates that are associated to it that you don't have to worry about. Specific scientists, you don't have to know their names. That's what the internet is for us to just do a quick Google search these days to find that out. But I just want you to be able to know the key discoveries, what scientific processes and technologies are associated with each era as you go along, and just have a very brief amount for each era. There's going to be a lot of information in here. I haven't had a chance to pare it down. Such is life. But I will highlight the key points for you, okay? So this is the breakdown that's in our slideshow, so you can take a peek at where we're headed. But this first thing is like coming up with a good definition for biotechnology. You've already had that opportunity to do that, but our government once had a sector that was associated to regulating and kind of leading and paving the way for biotechnology and what research was associated to it, that sort of thing. So they had an annual budget of around $22 million that they would, allocate to whatever they deemed important during this time frame. But their definition talks about any technique that uses living organisms or substances from those organisms to make or modify a product to improve plants or animals or to develop microorganisms for specific uses. So if I sum that up into two words, I would say something about genetic manipulation or being able to manipulate organisms or its components for the developing products for human needs and uses. So these are kind of the who for what's associated to biotechnology. But we can't forget about our little guys, the bacterias, because they actually play a really key role. And we can see all of the different areas that multidisciplinary areas that are associated to biotechnology in our sciences, but there are also other areas not directly related to science in which biotech um, would also be associated. um, so basically, why do we care about biotech? Why is this such a big deal? And you all are probably very well aware of this, but I still want to cover it, right? So our population is ever growing, right? We're somewhere around 7 billion. By 2050, our population is said to reach somewhere around 10 billion, which is insane. But we have this large population that's growing, and we need to make sure that we, still can support life with what's going on. So there has been this within the last 50, 60, 70 years, we call it the biotech boom or the green revolution, where there's been a huge increase in trying to understand and trying to develop various biotechnologies to try and improve human life. Our third world countries, looking at ways we can just make human life better and maybe the environment around us better too. So there are some examples here on how um, we've been able to kind of genetically manipulate or create some GMOs or virus resistant crops and plants, and even the same thing for our livestock, but creating various crops that have that virus resistance. We've been able to manipulate like the caloric outputs and specific nutrients that are associated to plants to make our lives easier. The other one down here that we kind of talked about as well are talking about our microorganisms or our bacteria. where we can manipulate their genetics and help them clean up Earth through waste and toxin removal, which is really cool. So there, and then the association to genetics, detection, and gene therapies are also really, really common for most recent biotechnologies. So our first era is called ancient biotechnology. I'm going to really shorten this up, and I know there's a lot of slides that are linked to this, but ancient biotechnology is early stages for our human population. And then thinking about, like when Egyptians were around and really early, early days that are associated to human life and the technologies that are associated to them. So the two main things that are going to be identified here um, is early farming, so early agriculture, and then early domestication of animals. So we originally were nomadic people moving from location to location, hunting, foraging, that sort of thing, and eventually got to this point where we could settle and start developing societies where it made more sense. And we were able to start collecting things and gathering um, our foods so that it made life easier. And by doing that, we were able to develop some early farming sites right on location so we wouldn't have to go searching for specific um, food anymore, and doing the same thing with domesticating certain animals so that we were able to utilize whatever they're producing to our advantage with like sheep and cattle and that sort of thing. So early farming and early agriculture um, is associated to ancient biotechnology. So again, why did we start settling down and becoming more sedentary? There's the examples here that I kind of referenced as well. And then once we settle down, this is when we have also early artificial selection. So no longer just letting nature choose its reproductive partners, but specifically looking at, creating certain, cattle that can have increased, whether it's related to meat or if we're talking about milk production, some specifics there, and doing some artificial selection so that we have the best of whatever it was that we wanted. And the same thing with our seeds and doing some genetic manipulation there so that we had certain um, changes to our crops and plants around us. Um, so here's an example, something that was found from some archaeologists, and you can literally see this for any seed and or plant. We have genetically modified them over time um, to be larger and to have different nutritional values to them than what they originally had. So this is early corn or maize, where it was really, really rough and difficult to actually get nutrients out of it. Like we have to seriously grind this thing up in order to um, get our nutritional value. Now we have the corn that's somewhere around 5, 6 times that size. It's softer. We have different versions of sweet corn um, that have some nutritional values that were not originally there with our primitive crops. And that's the same for anything. You could look it up. So ancient germplasm was also occurring during our ancient era. And germplasm is looking at the genetic um, information for all of our organisms, and then kind of thinking about ways we can um, save that information or create like banking, resource management um, centers in order to ensure that that genetic material is preserved, or we can still utilize it in order for future genetic manipulation. So one guy here, Nikolai Vavilov, um, headed up a very large scale um, genetic resource building. Um, and he started doing that kind of he was kind of the pioneer for that. And his values were in that fact of saying that we need this genetic information, and it's going to be important for us, not only historically, but for our future, if we can save the genetic information and seeds um, for all of our plants. And his values actually went against Stalin's values, and Stalin actually sentenced him to death and put him into prison because he didn't like what he was doing. But um, Nikolai Vavilov um, eventually has this, right? There's this giant resource of seeds that he started. We also have an early center, or one of our center storage is the National Seed Storage in Fort Collins. Um, and there's all sorts of different gene banks for not just plants, but other genetic stocks that are saved up. So here's Vavilov. Um, this is the earliest photo I could find of Fort Collins, and then kind of an updated version. It's still out of date, but kind of the banking system of how they stored the genetics information for all sorts of plants, and it's humongous in there. So, also in um, ancient era for biotechnology is early fermentation. At this time, we had no idea what was going on with fermentation, but we knew there was a process that allowed us to create certain foods like bread, wine, and cheese. And we didn't quite know this enzyme or microbe interaction part, but we knew that we were able to um, create these different products from um, some scientific processes or different techniques at this time. We don't really call them scientific processes. Eventually, um, understanding a little bit or making observations on this fermentation process. We have Pasteur being able to identify and create some baker's yeast from that. But again, there wasn't much understood from it, just knowing that we could make bread from it. So this is just early understanding and eventually developing some new techniques that go along with it. So the same thing for like cheese and understanding um, things like yogurt and butter. We can have different types if we can allow them to go through um, different processes. Adding different things to them leads to some diversity in our food. And there was a need for this because our diets were changing based off of the way that we were living. Here's an example of some early artifacts that are associated to baking. This is an example of large-scale production of cheese, um, and still associated to ancient era is fermented beverages as well. So um, again, that fermentation process, not very well known, but we did know we could make other beverages, maybe adult beverages that are associated to it by accident, right? So wine was left, grapes were left out and contaminated without really realizing it. And this delicious drink of wine was created that people just absolutely loved early on, but they really had no idea what it was that happened or how they did it. And they just kind of made observations and tried to replicate it again. Um, so here's an example, again, artifacts of huge vats of wine that have fermented over time. So our next era is classical biotechnology, and this is going to be getting into um, larger scale production of those previous topics we just talked about for fermentation and for various food um, creation. What should I say? Food science, right? So on the previous slide, it had Anton van Leeuwenhoek, who was able to actually visualize and see cells and microbes going through the fermentation process. So that allowed for the understanding that there are different types of fermentation. We don't really care about the specifics, but know that um, we started understanding some of the details because we could start developing some technologies that allowed us to see it in action. New products were being developed from this understanding or observation of fermentation. So, other things like glycerol, acetone, and butanol were understood and developed, and then eventually put into large scale production because of the wars that were happening around us. So, there was a big need for explosive devices. And so, large scale production or industrial fermentation was happening because wars were happening at that time. There was also an increased need for technologies and techniques that are associated to our healthcare system for sterilizing things and eventually developing antibiotics and vaccines um, during that time. So there was this mass production process associated to our products. And so that is associated to classical era of biotechnology. Just a funny sign I saw associated to penicillin. I'll link this into our assignment too, so you can watch it because a lot of things associated to science and our history and biotechnology actually happened accidentally, like we saw with the wine example earlier. Still with classical biotechnology, there were there was the capabilities and understandings of that there were things happening on a smaller scale that we couldn't quite see at this time, but we knew it was there. And so, looking at things like various compounds, the amino acids that lead to protein production um, and enzyme production, we were able to understand vitamins and start mass producing those. So, this was starting to get into um, more of the large scale commercial production pieces associated to biotechnology. Okay. Um, and just more examples that go along with that large scale production. And we'll kind of focus in on a few of these as we do our labs. So I'm just going to jump over it. Now we're in our modern biotechnology era, right? The last 50 to 70 years, like I said earlier, our biotech boom. Um, that's the modern era. And so the earliest is associated to microscopes and developing those technologies. So we're not just like seeing and observing science on a macro scale, but we could actually get in and see the cells and see what's happening at that point. So some compound microscopes were being developed. We now have electron microscopes that were developed too that allow us to see, not just cells, but we can actually see organelles and what they're doing. So these are early microscopes that were developed. For our modern era. The cell theory, we'll get into that, but it probably rings a bell as well. That was developed on how cells are the basic unit of life. All living things are made of cells, so they come from other cells. Um, and like I said, we could start looking at specific organelles and their functions. um, and just continuation of if we can look at cells and we can dive deeper, we can understand some metabolic pathways and some specific processes. So the modern era is focusing in on actually understanding each step-by-step process that's associated to like fermentation and um, our metabolic pathways and things like photosynthesis and that sort of thing, and being able to actually manipulate them to our advantage. So here's an example of Louis Pasteur and kind of saving food science and saving people's lives through the pasteurization process. Um, More technologies that are associated to it. Ultracentrifugation, which is that process of hyper spinning down your, not just blood, but you could do it with cell components, that sort of thing, and being able to separate out various components so you could really dive deeper into that. I already referenced the electron microscope and how that magnification allowed us to see more details along with it. Um, Understanding mitosis and meiosis and how cells reproduce became um, very well known in each step that was associated to it. And then eventually, sorry that went up there, but identifying DNA and genes, what they are, their structures, and their components, and all that. So here's Mendel's photo, and here's an example of an early electron microscope. And then we had our DNA kick, and that has to deal with our genetic manipulation that we now have capabilities over. And so understanding what is actually the driving component for genetic material, well, that's DNA. And so there were all sorts of experiments that were associated with that. Um, but we were able to say DNA, not protein, is associated to genetic material. And we can take DNA from one organism and we can bring it on over to another one. And that's called transformation. And we can start selecting specific genes that we want and move them from one thing to the next in order to get what we need. So here's an example of Griffith's early experiments that he did for transformation and moving genes from different organisms back when there was less concern about what happened to animals for scientific research. um, and so those processes continued with a whole bunch of different scientists being able to see and manipulate DNA and the genes that are associated to it. Okay. Um, all these scientists and their discoveries associated to the structure of DNA, allowing us to actually understand what DNA replication is and the repair process, and then being able to utilize that for some recombinant DNA or taking DNA and moving it to a different host cell or a different organism. So there's all sorts of different advancements that happened, but also then that led to the rise of bioethics and the need for something to potentially start regulating and kind of watching over what's happening in our biotechnology eras. So then, all sorts of debates arise associated to this DNA revolution, the DNA technologies that came along with it. But like I mentioned, eventually, um, biotechnology and all of these processes are deemed to help benefit human health. And so a lot of these debates eventually faded away because there was more benefit than harm happening. But there were many areas in which we made huge strides to make sure things were regulated. The CDC, the FDA, USDA, that sort of thing. They all have their toe in the water for biotechnology to make sure everything is safe and regulated and we don't have anything too wild happening. So that's kind of the breakdown associated to our eras. And then you can see some more details, but you don't have to know all of those. But just think ancient era is early farming, early domestication of animals, and fermentation that was happening without us really understanding what was going on, but new food and products were developed from that. Then our classical era is associated to um, being able to make observations on that macro scale and just um, recording those observations and then seeing how we can manipulate these processes of fermentation to create more products. We start having large scale production of biotechnologies for human needs, like those vaccines. And then our modern era is actually understanding the intricacies of each process and then therefore being able to manipulate what's going on with that.